For the science of evo-devo, it's really important that we understand phylogenetic relationships. And it's also really important that we understand how to read trees and how to map and look at characters on those trees. The methods for how we get our best theories, our best hypotheses about relationships is a little less important for the science of evo-devo, but I still want to review it. But with that said, we're going to do kind of a Cliff's Notes version. That's a little bit of a dated term. Cliff's Notes are short summaries of larger works like novels that you can use to kind of, I don't know if cheat is the right word, but kind of get a quicker view of, say, Pride and Prejudice rather than having to read the whole thing. You can read the Cliff's Notes version. So we're going to give you the Cliff's Notes version of phylogeny inference. How do we make a phylogeny? So you're not going to get all the details. If you've had my evolution class or perhaps another evolution class, you may have done this even in a little bit more detail. If you've had my molecular evolution class, which is an upper division course, we spend a good amount of time on this and you will already know this or at least have had it in the past in great detail. But for this class, this is the level that you need to know. So how do we make our phylogenies? Well, originally phylogenies were by and large best guesses. They were hypotheses, and they still are today, but the original hypotheses were based on not a lot of data. They were based on sometimes gut feelings, sometimes experience, which can get us somewhere if someone knows a lot about organisms, and even if they don't sit down and do a formal analysis or have a big data set, they could still get a lot of things right. One of the earliest written phylogenies that we have comes from Darwin's notebooks. He wrote, I think. He wrote number 1 down here as a root or common ancestor of all life on Earth, and then he just wrote a whole bunch A, B, C, and D, different branches representing different species. And he didn't do this as an intent to accurately reflect relationships among any group, just kind of to visualize his ideas about common ancestry. And so after theory of evolution becomes one of the foundational theories of evolution, or of science and biology in general, people began to try and figure out relationships. And they got a lot right just based on general morphological characteristics and experience and looking at things a lot and writing and drawing things out and writing things down. But in the 20th century, people began to say, hey, we need to have more subjective methods. We need to gather large data sets that can either support or refute our ideas about relationships among organisms. And so they began to gather data. And initially, all of these data sets were morphological. So does it have fur? Yes or no. Does it have a vertebra? Right? And many of these were already kind of intuitively understood, or large groups were already defined based on some of these major characters. But you quickly realize that sometimes one character can be a little misleading. So what we would like to do is gather all of the available data, all of the characters that help us to distinguish why one organism is different or the same as the other, and put those into a large data set that then somehow we could agree on how to analyze it and how to figure out what set of relationships best represents the historical connections between those organisms. Now, in the 1980s, 90s, going into the 2000s, we began to have the ability to sequence genetic data, to look at genes, and those genes became a new rich source of data for helping us understand phylogenies. So this one right here is a fairly simple A's and T's representing an adenine or a thymine at a certain position. And this is a data set. Now, there were originally, when people started to sequence data and gather data from morphology, there were two types of data sets. The first type of data set was originally, well, I don't know which one's first. I guess it depends on the type of data you're looking at, but we won't worry about which one had priority. The two types of data are discrete data, like DNA data, where you're either a thymine or an adenine. And then the other type of data was distance data. Now, distance data represents differences between different sequences or between different organisms. But rather than having a discrete, like, does it have a backbone, yes or no, you could do something on the order of how different are they from one another. And some of the original data, like protein migration rates in gels, were distance data. And they would be measured at how different is the protein from this organism versus this one. Is it, does it travel twice as fast in the gel? Does it travel a third as fast? What's going on? And so distance data is specifically different than discrete data. Okay, so just be aware of that. So in a distance matrix like this, I have the different species or sequences here, and I would compare 1 to 2 and say, there are 3 differences. I don't know what those differences are, but there are 3 differences between those. But between species 1 and 3, there are 5 differences. And so each of these represents how similar. If we have a 0 there, then that means those 2 sequences are identical to one another. Okay? And so there are different methods that are used to analyze discrete data versus distance data. Now, our large data sets today, which are made up primarily of molecular data from genes, are discrete. And there are 4 different, well, actually technically 5 different possible character states for any one position in the gene. You could have an adenine, a guanine, a thymine, or a cytosine, right? So those are the 4 base pairs. Or you could have what's called a gap. And a gap represents an insertion or a deletion, a mutation. So those are the 5 different things you can have in an alignment of a DNA. This is one from a protein coding gene for some frogs that I did for another class a while back. But I just want to get you an idea, and just you should know that genetic data is discrete data. Now, discrete data could be transformed into distance data if we wanted to do that. And some early approaches to making phylogenies did that. And so if we wanted to change this to a distance matrix, all we would need to do is say compare the first species to the second species and count up all the differences. Oh, there's a difference there. There's a difference there. There are 3 differences. We count up all the differences, and that becomes a distance measure for how different those species are from one another. And we do that for all of the pairwise comparisons, so 15 versus all of them, 16 versus all of them, and so on, until you've done all pairwise comparisons. And you could generate a distance matrix. It would be much larger than this one because this one only has 4 sequences in it, all comparisons between those 4 species. Okay, so just know the differences. Now, originally, there were large amounts of effort and energy that went into both methods for doing distance data and methods for analyzing discrete data, and then trying to determine what the best set of relationships was based on that data set. By and large, distance measures have been replaced by the discrete measures. And so we won't talk about many. We're going to do one distance measure, and it's used not as an end of the analysis, but as a tool to help us do better analyses for the more complex and intense analyses that we do for discrete data. So just know that distance data was important early on. It's not important as an end result anymore, but it is important as a tool. One of the main advantages of discrete data over distance data is that we can infer actual evolutionary changes when we are using the discrete data itself. So parsimony is one discrete method. And via parsimony, we won't get into this in much detail, but you can map the actual changes onto your phylogeny. So here we've got an unrooted phylogeny showing relationships between 4 species. And we mark specific changes. So character number 6 has a mutation right here. That means 3 is different than everything else. Character 5 has a mutation on this branch, which means 3 and 4 are the same, but they are different, separated by that mutation from species 1 and 2. Whereas with distance data, although we can say how many changes occur maybe along any one branch, we cannot say what those exact changes are unless we go back to the original discrete matrix. And some data didn't have discrete matrices at all. So like electrophoresis, or if you're measuring absolute size, is the average size of the length of the femur of that species X centimeters long or Y centimeters. Those are all distance measurements, and you can't go from distance back to a discrete matrix. You can go from discrete to distance, but once you do that, you lose some of the information, like what changes are occurring. So that's the main advantage of discrete data, and by and large, we've decided that advantage far outweighs any reason to do distance analyses when discrete data, like large DNA data sets, is available. Okay, so here's the only distance method we're going to talk about. It's called neighbor joining. And it's a very efficient method. And what I mean by efficient, when we're talking efficiency in molecular phylogenetics, we're talking about how much time, how much computing power, which is really just a measurement of time, right? If you have the same computer doing two different problems, the more efficient one would be the one that that computer can do more quickly. Okay, so it's a very efficient method, but it has the drawbacks of distance methods. And so we don't usually use neighbor joining nowadays as the final result of an analysis. However, it is a tool, a starting place for these more complex analyses. And so here we have a neighbor joining tree showing data from some primate species, humans, chimpanzees, gorillas, gibbons, with information representing, and I think this is, what are the chances of any single position in the matrix having a mutation? So 5% of positions have a mutation between chimpanzee and the common ancestor. And if we add up the distances between here, we can get estimates of how many total changes there are in this gene between chimpanzees and humans, or between chimpanzees and gorillas, or any of them. So there is some information, but again, at what position in the gene those changes are occurring is not available with this distance method. So neighbor joining is an efficient method to get us in the ballpark, a pretty good estimate of actual final relationships. But we then use that as a tool for the next stage of our analyses. So you should be able to make a distinction between distance methods and discrete methods. And then there is a second kind of dichotomy in phylogenetic methods that I also want you to know. And that is between what we will call clustering methods, sometimes called algorithmic, but we'll call them clustering methods, and optimality methods. A clustering method is a method that, rather than trying to find the very, very best overall solution based on some criteria, you just have a set of instructions or an algorithm. You do A, B, C, and D, and you get your end result. And the end result is not guaranteed to be the best overall tree based on any criterion, but it's going to kind of get you in the ballpark. Okay? Now, an optimality criteria ideally searches every possible solution and gives you the best overall solution. So I like the analogy. An optimality criteria is a lot like the apps that we have on our phone now to get us to a different location. Right? So if you wanted to drive, especially in a big city, if you wanted to drive from one location to another one, you could get on Google Maps or any of these like Apple Maps, and you can put in an address, say, I'm here, here's where I want to go, tell me how to get there. And the algorithm runs through a number of different characteristics, and it gets you the optimal way to get there. The fastest way, I think, is the default. But I don't know if you've ever done this on a map. You can actually change the optimal criteria you're looking for. Let's say you wanted to find the one that traveled the least distance in miles, and that might be different. You know, maybe you take surface streets instead of the freeway, and you can get there using less miles on your odometer, but it might be slower because on the surface streets you have to stop at all the lights. And so an optimal time versus an optimal distance might be different solutions. Okay, but that's, you think about it, it's a really complex problem. How many different ways are there to get from point A to point B in a large city? Right? If you're traveling between cities, you know, usually there's only one major highway or freeway to take, and then a little bit of small choices when you get to the city. You know, which exit do I get off on? You know, do I take this belt route or this one when I get into the city? There might be some other changes, but within a city, certainly there are, maybe, if you want it to be extreme, there are billions of ways to do it, right? So just as an example, and this will be important later. Let's say you wanted to travel from McAllen to San Antonio, okay? And obviously, there's going to be many, many different ways to get there. And you could even be really, really extreme, right? You could say, well, if I want to get to San Antonio, I'm going to cross over the border and get on an airplane in Matamoros, and then I'm going to fly to San Antonio. And that gets you there, and it might even be, you know, a pretty fast way to get there, but maybe you're looking at efficiency based on cost, right? It's probably a more costly way to get there than just simply driving up 281 and then, you know, taking, I think it's 35 or whatever it is to San Antonio, right? So realize there are many different ways, and some of them are pretty extreme. You could say, I don't want anyone to find me, so I'm going to get an ATV and go off-roading and cut down people's fences on their ranch and drive all the way up cross country without any major roads. You could do that, but that's a pretty extreme way to get there. So realize there are many, many different ways. Most of the time we go based on time or cost, and we try to optimize one of these criteria. So with that in mind, now let's apply this to phylogenies. So neighbor joining uses distance data. This is what we talked about earlier. And it's a clustering algorithm, meaning you do A, B, C, and D. You're not guaranteed to get the very best answer, but you're going to get kind of in the ballpark in the neighborhood, right? There are other distance methods. For the purposes of this class, we won't worry about them. You don't need to know about UPGMA or minimum evolution. Don't worry about those, okay? Now, the other methods that we are going to talk about are parsimony, maximum likelihood, and Bayesian analysis. Now, both parsimony and maximum likelihood are very clearly discrete methods. They use nucleotide sites, which is a type of discrete data, and they have an optimality criterion. Now, the criteria that they are optimizing is different. Just like we might do it based on time versus cost for, a trip, we could do it. And parsimony tries to minimize the number of overall mutations, where maximum likelihood tries to minimize a score based on a statistical analysis of the data. And for this class, we're not going to get into the mechanisms or the details of either of those. And you don't even need to know what criteria they're trying to optimize, as long as you know that they're both optimality criteria. Okay. Bayesian analysis is a little bit less clear as to what it's doing. Some people would argue for it as an optimality criterion. Some people would argue it as more of a clustering algorithm or a hybrid of the two. That's why I put a question mark by it. But it does start with the original genetic data. It's not a distance method. It uses the discrete data. Okay, so let's talk just a little bit more detail about these methods, and then we'll kind of put a pin in our methodologies of how we build trees and say that's good enough for the Evo-Devo course. So we're going to start with parsimony. Parsimony minimizes the number of homoplasies. Now, a homoplasie is when we have to infer a change has occurred twice. So let me give you an example. This is kind of a silly example, or maybe a simplistic example, but it illustrates the point well. And sometimes simple examples are the best ways to illustrate a point. Let's say you wanted to figure out how a penguin, an eagle, and a bat were related to one another. Okay. Now you'd say, oh, we're going to sequence big data set, lots of genes. We're going to do an alignment. We're going to do an optimality criterion and study lots of different trees, and we'll find the best one. Okay, great. And you could do that. But let's just illustrate it like with a couple characteristics. And rather than genetic data, just because we don't know it offhand just by looking at them, let's use some morphological characters that we can figure out just by looking at these critters, right? So you might look at them and initially say, oh, the penguin and the eagle are birds, so I'm going to put them together, and the bat's a mammal. We'll put them more distantly related. And that's a great way to do it because those taxonomic groups, right, those classifications are based on these understandings. And why we call it a bird is because it shares all of these characteristics, okay? So they've got feathers, they have a beak, they lay eggs, and you could do there are a number of really, really good bird characteristics that a mammal doesn't have, okay? So that would work. But realize that if you didn't know these groups very well, there might be some characteristics that were misleading. So for instance, the eagle and the bat both fly and have this kind of general forelimb shape that allows that way for them to move through the water, where the bird wing in the penguin has been modified greatly. It doesn't look a lot like other bird wings. So if we did that, if we use that as a character, then we would tend to put the eagle and the bat together because they can both fly. And then along this branch somewhere here, we'd mark, oh, look, flight only originated one time after the penguin broke off from these two groups. And the reason that the eagle and the bat can both fly is because of their ancient ancestry evolving that ability to fly. We mark it once, boom, we're done. Well, this is why we don't use single characters, right? If we added characters to there, if we said, oh, let's look at feathers, you'd say, oh, wait, the bird has both of the birds, the eagle and the penguin have feathers. And you would have to mark feathers twice. That would be a homoplasie. You would have to mark the beak evolving twice, right? You would have to mark eggs evolving twice. And we could mark them either as convergence, right? The egg evolved twice here and here, or the egg evolved in an ancestor and then was lost in the mammal. That would be a simplesiomorphy instead of convergence. But both of them are forms of homoplasie. And as we gather characters, we realize pretty quickly that this set of relationships is a bad one because we have to mark so many characteristics, these bird characteristics, we have to mark them on there twice. And so we would much prefer instead, let's make the change here, right? A better set of relationships is that the penguin and the eagle are more closely related to one another. And the bat is distantly related. And we'd have to mark the origin of flight on there twice, right? But all the bird characteristics now we can mark in a single place. And so that is a better solution. The optimal tree is the one that is minimizing those number of homoplasies. Okay, that's phylogeny using parsimony in a very quick one. And I'm not going to make you build trees, but you should be able to recognize characters using that criteria of, okay, I'm going to try and make them occur the least number of places on a tree. You should be able to represent what a synapomorphy is versus the two types of homoplasy. So we did that in the last lecture. Review it or log on with me if you need to review it. All right? So that's a great method. And by and large, we've decided that this is a better method for making our phylogenies, but it does have drawbacks. The main drawback to an optimality criterion like parsimony is that it is very inefficient. It takes a lot of time or a lot of computational resources if you want to think about it that way. So the problem with optimality is that it is a massive number of possible solutions. If you wanted to evaluate every single possible tree, the number gets really big really fast. And this is the equation for how you figure out how many trees you would have to evaluate. Many of you are not familiar with this type of operation in scientific notation. It doesn't matter. I don't care that you memorize it. What you do need to know is that it's a huge number. If we have 3 species, there are only 3 ways to organize them. There are 3 possible phylogenies. But notice that as we're going up, adding 1 species to our analyses, this number gets bigger and bigger and bigger and bigger. By the time we have somewhere in the neighborhood of 50 species, there are approximately the same number of possible phylogenies as there are atoms in the visible universe. It's just an incomprehensibly large number. Even our best computers cannot evaluate every single possible tree. And so we're left with this little bit of catch-22. We want to do optimality criteria like parsimony, like a maximum likelihood analysis. We'd like to use those methods, but we can't evaluate every possible solution. So are we stuck? How do we work our way out of this paradox, this catch-22? And I'm going to give you an analogy here with the trees, and then we'll come back to our methods for getting from one place to another as an analogy. So it turns out that if we were to map all possible trees based on their score, with the best trees being kind of in these high places, this is just a small set of solutions, but we would have a very, very different topology here if we had many, many species in our analysis, that there is a small number of trees that are going to have close to the very best score. And there might be 1 or 2 or 3 trees that have the very best score overall, but there's a small number of trees that have the best score. And so we maybe don't need to evaluate every single possible tree to be relatively certain that we found the best score. So let's make an analogy back to our mapping app on our phones. Let's say you want to go from here to San Antonio. And the algorithm runs through a number of different solutions and spits out a possible solution to get you there maybe the fastest way possible. And you're like, oh, wait, but you didn't, and if it really is the fastest way possible, right, then maybe your best solution is to jump on a charter jet and fly to San Antonio. It's not a very cost-effective method, but it would be based on time. But if somebody came to you and said, oh, wait, you're looking for the fastest way possible, you didn't check out if I got into a kayak and paddled down the canal and then, sprinted to the nearest street and then called an Uber. And, this really obviously ridiculous. You say, well, I don't even need to spend time evaluating that method because it's obvious that it's not going to give us a good solution based on the criteria of getting there quickly. So there are lots of solutions in phylogenies, these big phylogenies, that we really don't need to evaluate because they're so far from the best solution that maybe we can look at a couple trees that are a little bit like that and decide, oh, those are really bad scores, so we're not even going to spend any more time looking at trees like that. And so we can get around this catch-22 of not being able to evaluate every single possible outcome by using some kind of smart search strategies, right? So we don't need to search every tree. We can search a subset and be reasonably certain that we found the very best overall solution based on the criteria we're using. Now, the last two optimality criteria, or at least discrete criteria, may be optimality depending on how you define Bayesian analysis, that I want to talk about are maximum likelihood and Bayesian analyses. Okay? I've put these big scary equations here just to freak you out a little bit. Don't worry about them. I just wanted to illustrate basically the take-home message for this, the level of complexity that you need to know, is that maximum likelihood was a way to statistically analyze the data and minimize a statistical score to find the very best tree overall. And it uses some information inherent in molecular data sets, like how many adenines are there versus guanines. How often do I see an adenine changing into a guanine? And to make some of those measurements, we need a starting tree, which is where our neighbor joining tree comes in. So when we do a maximum likelihood analysis, we do a quick neighbor joining tree to get a good starting point. And then we're able to make some inferences about the data set on that phylogeny to come up with our values for our statistical analysis. And we run it through this big, crazy, unless you're a statistics major, you're going to look at something like this and say, I have no idea what even a little bit of that means, which is fine. But you know that we take our data, we analyze it, and we run it through an analysis. Now, the problem with that, many people begin to say, oh, this is a better way to do it. But the problem with it is it takes 10, 20, 30 times as long to analyze a single tree using this statistical method as it does in a simple parsimony analysis. So we've kind of compounded our problem of optimality and finding the best tree overall once we begin to use statistical methods like maximum likelihood. To get around that, people eventually invented a Bayesian method, which is a subclass of statistics, but they invented a Bayesian method to analyze these big data sets. So take-home message, nutshell version, Bayesian is a way to use statistical approaches, statistical data from the data itself, and to find a best overall tree, but to do it in a more efficient manner. And so today, most analyses involve you doing 1 or 2, 3 methods and then comparing the trees. We might do a parsimony method, we might do a Bayesian method, and then see if the trees are similar or different. If they're much the same, we can be more confident maybe in our solution. But many of the large analyses you'll read will be based on Bayesian methods. That's a very common method today. And it's kind of, I don't know if you could say the best of all worlds, but you could say it makes some compromises between all of these different criteria. It's a relatively efficient method where we can use the discrete data and we can do a statistical analysis of our data and we can find the best overall data set. And today, people are working on many, many different groups using these modern methods, adding more genes, adding more data. And for many groups, we're becoming very confident. Our theories about relationships among organisms in many groups are beginning to get to the area of, our hypotheses about these relationships, right? Our initial ideas based on a preliminary data set are beginning to become more like theories, where we have lots and lots and lots of analyses, lots of data that are supporting the same result over and over and over again. And some of these results were what we already knew. We knew mammals were all a single monophyletic group, and all of the genetic data has supported that. But others we are revising some previously looked at groups. And depending on the group you're looking at, we might have had some major revisions over the last couple decades based on all of this genetic data that is coming in. And although this is an ongoing process, many of our phylogenetic relationships are pretty well, or maybe even very well, figured out, and we know them to a great degree.